Good evening to all the CUET aspirants. So here we are with the chapter Phycology. So far we have completed Microbial Diversity 1, where completely we have dealt with bacteriology and virology by exercising few CUET-PG questions. Today in this session we are going to deal with Phycology, wherein we are going to start with algae, its general characters, structure, reproduction, classification. Especially we are going to follow Fritsch classification, and then cyanobacteria, general characters, cell structure, organization, their role as biofertilizers. Examples if you see, Oscillatoria, nostoc and anabina, we are going to learn detail about this. Followed by Chlorophyceae, Phaeophyceae and Rhodophyceae, and these type studies we are going to learn it in detail. This is the complete gist that we are going to learn from Phycology. Clear, right? Now, first look into the general characteristics of algae. Algae, which are known as primitive plants. Okay, these are primitive plants. Once let us recall all the 5 divisions of plant kingdom. Plant kingdom has 5 divisions. Okay, kingdom Plantae, has 5 divisions. Okay, they have kingdom Plantae has 5 divisions on total. Okay, first will be Thallophyta, thalloid organisms, which include algae. So these are primitive plants. Second is bryophytes, followed by pteridophytes, followed by gymnosperms and angiosperms. Fine, right? So we have Thallophyta, Bryophyta, Pteridophyta, gymnosperms and angiosperms. First, let's get started with Thallophyta with their general characteristics. If you clearly look into the general characteristics, the age of algae is around 4.5 billion years old. The algae appeared as early as 3.5 billion years ago. Algae has appeared 3.5 billion years ago in pre-Cambrian era. Okay, they have appeared in pre-Cambrian era. First photosynthetic organisms. What are the first photosynthetic organisms? Algae are the first photosynthetic species on the earth, and they have crucial role in the oxygenation. Algae were responsible for releasing oxygen into the atmosphere as they are conducting what type of photosynthesis? Oxygenic photosynthesis. They do conduct oxygenic photosynthesis. Oxygenic photosynthesis. Fine, right? Where this is being releasing oxygen into the environment for the evolution of life. Next, aquatic dominance. Algae dominates around 3/4 of the earth's surface. Fine, right? Algae dominates around 3/4 of earth's surface. They are known as seaweeds. Next, fossil algae, most important. Stromatolites, fossil forms, these are found in Shark Bay and Western Australia. And oncolytes, these are oval or spherical shaped structures which are found in Kadapa basins of India. Next, study of algae. Okay, study of algae. Just go through this. Yeah. Study of algae is addressed as phycology or algology. This is derived from Greek word which means phycos or seaweeds. Fine, right? This is the basic introductory part of algae. Now if we move on to the characterization of or the characteristics starting from the habitat. Okay. Firstly, algae are widely distributed in the aquatic habitat. They are distributed both in the water and the marine water habitats. They are also found in moist soils, over tree trunks, in snow and on wet walls. Their adaptability and habitats. Based on their adaptability, the algae can be used floating phytoplankton, okay, floating algae, phytoplankton, dwelling algae which are known as benthics, okay, those which are attached to rocks, those are known as epilithic, those which are attached to soil particles, those are known as epiphilic, okay, and those which are present on the surface of other animals which is known as epizoic, okay. Remember, those floating algal species are known as what? Phytoplanktons or plankton species. Next, bottom dwelling on the rocks, those are epilithic, attached to soil particles, epiphilic, and on the surface of the animals, it is known as epizoic. Best example is sloth bear. Sloth bear gets camouflage activity over sloth bear when algae is completely covering the sloth bear. The surface of the algae is completely covered by sloth bear, which gives camouflage effect, and sloth bear will be escaped from the predators. Sloth bear is escaped from predators. Okay, sloth bear is escaped from predators by the camouflage activity that is provided by the algal species. So, symbiotic association, you can see that their association can be with protozoans, with coelentrates and with mollusks. If algae is present on the surface of animals, that association is known as epizoic. If they are present inside the animals, that association is known as endozoic. If that is inside the plants, it is known as endophytic. Fine, right? Epizoic, endozoic and endophytic. Clear till here? Any doubt anywhere, ma? Yeah. This is about the habitat of algae. Now, lichen formation. We know lichens are the pioneer species of ecological succession. Okay, lichens are the pioneer species which starts up the process of succession. Few algae form lichens by forming a symbiotic association with fungi. Okay, algae, when they form symbiotic association with fungi, okay, they form what? Lichens. And also we have parasitic algae like Cephalourus species. Cephalourus species is a parasitic green algae which is on the angiosperms like anacardium and rhododendron and thea. These are the plants on which Cephalourus is parasitic on. Remember, very important about parasitic algae. Next is algal blooms. Some algae will form blooms in large number. Okay, algae will form blooms in large number. Algal blooms are also addressed as water blooms. Examples you can see Microcystis and Gymnodium and Goniolax. These are the examples for what? Algal blooms or water blooms. Goniolax in specific, it is a dinoflagellate as it is unicellular species. As the unicellular species, they are addressed as dinoflagellates which are single celled protists. But then they also perform photosynthesis. They have algal like characters. So this is also included under algae. Fine, right? So Microcystis, Gymnodium and Goniolax, these are forming what? Algal blooms. Next is about thermophilic algae. Thermophilic algae, some algae grown in what? Hot springs at 70 degrees Celsius. Some algae grown at 70 degrees Celsius. For example, Syncocystis. Okay, this is the example for what? Thermophilic algae which lives in the hot springs, whose habitat is hot springs. Clear till here? Lichen formation, algae as parasitic algae, and about algal blooms and thermophilic algae. Next. Structure of algae. Yeah, structure of algae. The body structure of algae has simple plant body. Algae will have a simple plant body, that is thalloid plant body, which is thallus like. Thallus has undifferentiated body. Thallus is what? Undifferentiated body, where there is no root, stem or leaves. And comparison of this with bryophytes. Bryophytes with thallus. Some bryophytes also have thalloid bodies. Okay, some bryophytes will also have thalloid bodies. For example, Riccia, Marchantia and Anthocerus. This is the comparison of few bryophytes with algae. Sex organ differences. Bryophytes female sex organ is archegonia and it is multicellular, whereas algal sex organs will be unicellular and all the cells will be fertile in the algae. Clear till here? Any doubt anywhere, ma? Yeah. Now we will move on to diversity of thallus in algae. Okay, diversity of thallus in algae. Thallus ranges of body forms. Algal thalli range from microscopic, that is micromonas, to giant forms like macrocystis and neurocystis, up to 108 meters. Up to 108 meters. They range from microscopic to giant forms. Types of thallus, if you see, we have unicellular thallus like Chlamydomonas, Cenobium like Volvox, filamentous like Edogonium, heterotrichous like Chilocheta, and laminate or leafy forms like Ulva, xenophous forms like Enteromorpha, and those algae which has root, stem and leaf like parts like brown algae, if you see, example is Sargassum. Sargassum is the example. Clear? Next, vascular tissues. Algae lack true vascular tissues, but some conducting substances will be present, but there is no true vascular tissues like xylem and phloem. Clear? This is the diversity of thallus. What is that important in this slide that you have to remember for your exam point of view is the macroscopic algae and different thallus structures with their examples is very important for your CUET. Okay, example based. Complete core botany chapters, be it phycology, mycology, archaegonia, bryophytes, pteridophytes, gymnosperms and angiosperms, until embryology of angiosperms, which are core botany chapters. What is that most important in this is memory based questions. Complete memory based questions you will be getting it. Clear, right? Now, types of thalli based on motile habitat. Types of thalli. Motile habitats here, unicellular forms, cenobial forms, colonial forms. Motile habitats, unicellular forms, you see the simplest thallus in the algae. This is the simplest thallus in the algae. Cells are motile. They are 1 to 4 flagella. They are present over here. First is Chlamydomonas species as Chlorophyceae. Heterochloris of Xanthophyceae, Chromulina, Chrysophyceae, Euglena, Euglenophysii, Cryptomonas of Cryptophyceae, Sodium of Dinophyceae. These are different classes with the species example. Now, unicellular forms, different species and the classes they belong to. Cenobial form, if you see, that with unicellular form. Now we will come across cenobial form, colonial form and cocoid form. Cenobial form, these are bounded by some mucilaginous sheaths. For example, if you see Gonium and Pandorina, which belongs to which class, ma? Chlorophyceae. Cyanura, Chrysophyceae. Fine, right. Examples under each thallus form is important. Colonial forms, if you see, of course, Volvox, and Eudorina also you can take of chlorophyce only. Next, coccoid forms. These two sets of examples, I hope this is clear for you. Next, coccoid forms, we have two types again, unicellular and coenobial forms. Okay, we have again unicellular and coenobial forms. Unicellular forms examples, if you see, chlorella, which belongs to chlorophyce, Botrydopsis, which belongs to xanthophyce, Chrysophora, which belongs to Chrysophyce, Tetragonidium, Cryptophyce, Cystodinium, Dinophyce. Okay, these are the examples of unicellular forms. And coenobial, and these two are the examples. Coenobial, where the cellular arrangement is in the single row, Kenidesmus, and if there is any net or hexagon or pentagon type of coenobial arrangement, example is Hydrodictyon. All the classes or the habitat types, the examples are important both for motile and non-motile forms, unicellular and coenobial, motile and non-motile forms, and coccoid habitat is completed so far. Now we have to look into palmar and dendroid habitat. Species which lose flagella in vegetative stage, they possess them in reproductive stage only. Okay, so species, what type of species are present? They lose flagella in vegetative stage, only in the reproductive stage the flagella is present. Okay, major point to be addressed for palmaloid habitat. And what type of cells you can see here? Non-motile cells which are embedded in a mucilaginous sheath. Okay, all the cells are non-motile, they are embedded in a mucilaginous sheath. Examples you see, Microcystis of Psilosporium of globular tubular and Eucapsis alpina. These are the examples of palmaloid habitat. Next, dendroid habitat. In this thallus, every cell base is gelatinized. Every cell base is gelatinized. Due to gelatinization, polarity of cell is well differentiated. Polarity of cell is well differentiated. Cells will possess stalks at the base, and these species are branched in structure. Okay, they are branched in structure. Examples, if you see, Cynostyron, this is example, and Ecdysocystosis is the example for chlorophyce. All the examples are important, ma. Have screenshots of this, revise it multiple times. Next is filamentous habitat. Filamentous habitat, this occurs in the organisms which exhibit cell division in a single transverse plate. Okay. So when cell division happens in single transverse plate like this, the cells are appearing in what? Transverse plane, which forms single. Each plant body is termed as filamentous form. Filamentous form, again you have unbranched filamentous form and branched filamentous forms. Unbranched filamentous form, single row of algae which is uniseriate. In some cases, cells filament will divide and arrange. They are dividing and they arrange in multiple rows. They will be arranged in multiple rows, which is addressed as multiseriate forms in the same plane. Example, you see Ignima and Spirogyra, which is having seriate or single row. Schizopus of multiseriate type, it belongs to the class Chlorophyce. Fine. Next, branched type of filamentous form, branched filamentous forms. In many algal species, filamentous forms will exhibit branching. They exhibit branching, and branches will arise from apical cell. They arise from apical cell or many other cells of the filament. Examples you see, Cladophora of Chlorophyce, Monocelia of Xanthomonas, and Phyothamnion of Charophyce. Fine, right? These are the examples of branched filamentous forms, and these are the examples of unbranched filamentous forms, where you will see both uniseriate and multiseriate type. Clear till here, ma? Any doubt anywhere? Next. filamentous habitat, false branching and heterotrichous habitat. False branches, it is found in some members of Cynophyceae. Fine, right, it is found in some members of Cynophyceae where filaments will protrude out of the mucilaginous sheath. Here false branches, filaments will protrude out of the mucilaginous sheath which secrete out the new sheaths. Example is Chaetonema. Okay, the filaments will protrude out of the mucilaginous sheath like this. For example, Chaetonema. And last type of habit that we are learning, last type of thallus form we are learning, that is of heterotrichous habit. Heterotrichous habit which is with two distinct filamentous habitat. It is with two distinct filamentous regions. One is prostrate system and second will be erectus system. Prostrate system, horizontal growth is attached to the substratum. Horizontal growth is attached to the substratum here. Erectus system, where vertical branches arise from the prostrate system. Prostrate system, this shows horizontal growthing. But here in erectus system, vertical branches will arise from the horizontal growth. Fine, right? Example is Chilochaete of Chlorophyceae. In others, erectus system is more decayed like Drapanaldea and Drapanaldeopsis. These are the examples for the erectus system in other species. In Frexilla tuberosa, both systems are well developed, which resembles a small plant. Fine, right? So these are the two species. This filamentous habit and next is heterotrichous habit. You can take a screenshot of this. We have two more habits. One is siphonaceous habit, where vegetative growth is well developed but this lacks some cross walls due to what? Absence of cell division during growth. Cell division is absent. This will also result in cenocytic condition or multinucleated condition. Okay, cenocytic or multinucleated condition where cell walls will appear only during reproduction and injury. Okay, cell walls will appear only during reproduction and injury.
Really good.
Specialized
Hominis
large central vacuole
are there. Remove them. Cells happen
here. Examples, if you see protosiphon, which is
the excess
and rhizoid ground part is spread.
double bond
And ciliary, which is uninucleated vegetative cell and multinucleated reproductive stage is seen. And caulerpa is differentiated into rhizome, similatory filaments and rhizoids. Botredium and Wucheria. Which is siphonous forms in xanthoforms. Which forms? Siphonaceous forms in xanthophyceae. Clear with all these examples of siphonaceous habit. Siphonaceous habit feature is multinucleated or cenocytic condition. Fine, right? Is it? Is the sound clear now, ma? Is the audio clear now?
hydrogen.
Is the audio clear now? Last thallus habit here is parenchymatous habit. This is believed to evolve from filamentous forms, where cells will divide in all planes. Cells divide in all planes here, which form a parenchymatous thallus. Okay, complete parenchymatous thallus is formed. This parenchymatous habit is of two types again. One, it can be in the form of tubular habit or it also can be in the form of foliose habit. Tubular habit and foliose habit. Tubular habit, these are found in what type of species? Monostroma species and enteromorpha species of chlorophycean class. Monostroma and enterostroma species. Thallus is single celled in thickness and this resembles tube. Hence this is also known as what type of thallus? Tubular thallus. Fine, right? Next, foliose thallus. Thallus cells will divide into two planes here.
It's
Both anticlinal divisions,
HN
periclinal divisions. Fine, right? So thallus cells divide in both the planes to form 2 to 3 cell thick structures. Example is dictyota. Fine, right? Fucus here, fucus which is the brown algae which has diplantic life cycle. This resembles what? Leaf-like thallus. In members of Phaeophyceae, for example, laminariales and fucales, thallus is well developed and this is differentiated in
water molecule is removed by hydrodex
and other
nitrogen
meristematic tissues. For example, sargassum. Other meristematic tissues like sargassum. Clear till here? Any doubt anywhere? Here we go with cell structure. Okay. Two types of cell structures are seen in algae. Prokaryotic cell is observed or found in cyanophycean cell or prochlorophyceae, which are primitive algae again. These are primitive algal members. Okay, they are found in cyanophyceae and prochlorophyceae. They lack organized nucleus and a nuclear membrane. They don't have an organized nucleus and they don't have a nuclear membrane. No nucleoli, chloroplast, mitochondria and endoplasmic reticulum, because the cell represents a prokaryotic cell. But then it conducts photosynthesis, so these are placed in the algae. Fine, right? The cell characteristics are of prokaryotic cell, but the photosynthetic nature is making it an algae, unicellular algae. Cell wall is composed of mucopeptides and muramic acid. Example for this is what? Cyanobacteria. Okay, nostoc, anabina, oscillatoria, these are few cyanobacterian members. And what are the cellular characteristics of that? These are the cellular characteristics. Not a well developed or well defined nucleus, no nuclear membrane, no membrane bound organelles are present, and the cells are covered by a mucilaginous coat, and then cell wall is of what? Mucopeptides and muramic acid. Next, eukaryotic cell. Nucleus is bounded by a nuclear membrane here. Organelles like chloroplast, mitochondria and endoplasmic reticulum, these are membrane bounded. Okay, they are membrane bounded. Next, cell wall in algae. Cell wall in algae, we have, algae will show many variations like unicellular flagellates where cell wall is incomplete here. Unicellular flagellates where cell wall is incomplete. Prokaryotes and eukaryotes, cell wall is complete here. Okay. And cryptophyceae, xanthophyceae, chrysophyceae, here cryptophyceae, xanthophyceae and chrysophyceae, where cell wall may be absent. Remember, every point is important over here. If you want, you can take a screenshot of this and revise each slide multiple times, because all the statements here or all the examples which are given out here in phycology or in core botany chapters is purely memory based. Okay. Instead, cytoplasm is covered by pellicle, periplast and genus lorica. Next, chemical nature of algal cell walls. What is the chemical nature of algal cell walls? It is made up of cellulose, mucopolymers and silica. Structure, if you see, cell wall is composed of polysaccharides, cellulose and pectin. Cellulose forms inner layer with microfibrils. Pectin is forming outer layer in structure. So, cell wall of algae, most of the algal species, it has two wall layer components. Outer wall layer is made up of pectin, and inner wall layer is made up of cellulose. Fine, right? Outer wall layer is made up of pectin and inner wall layer is made up of cellulose. Clear, right, ma? Next. Yeah. Here we go with algal cell wall composition. Algal cell wall composition. Chlorophyce, xanthophyce. Here you see outer layer is amorphous, which dissolves in water forming mucilage. Okay, this dissolves in water forming mucilage. Okay, rhodophyce, phaeophyce and dinophyce. Okay, inner wall is cellulosic and outer walls will have xylenes, galactanes and pectins. This is observed in chlorophyce and xanthophyce. In phaeophycean members, the outer wall layer contains of or consists of carrageen, alginic acid, fucoidin and fusin. Alginic acid will form salts. Alginic acid forms salts with sodium, potassium, magnesium. These are commercially called as what? Alginates. They are commercially called as alginates. Right. Now, bacillariophyce and chrysophyce, where the cell wall contains silicon dioxide. Cell wall here contains what? Silicon dioxide. Now, cyanophycean species. Cell walls have mucopeptide and muramic acid. Plasmodesmata are seen in rhodophyce and cyanophyce. Diatoms and desmites, they show small apertures in the cell wall. These are key characteristic features or the components that are present in different classes of algae. Okay, the outer cell wall components or the cell wall composition that is present in different algal species of different classes. You can take a screenshot of this, very important slide.
Yeah.
Next. Pigmentation is one of the major criteria for classification of algae. Okay, as such we have three major types of pigments that are present in the algae. One is the set of chlorophylls, carotenoids and phycobilins, which are bilin proteins. These phycobilins are present in mostly rhodophycean species. Here we go with solubility. Both chlorophylls and carotenoids, they are water insoluble. They are soluble only in the organic solvents, whereas phycobilins, both phycocyanin and phycoerythrin. Phycobilins include all the phycocyanins and phycoerythrins. Phycocyanins and phycoerythrins, they are present in, they are water soluble. Bilin proteins are water soluble, whereas these are water insoluble. Clear, right? Now you see different chlorophyll types. Chlorophyll A is observed in all the algal species. Chlorophyll B is seen in chlorophyce, euglenophyce and paracyanophyce. Okay, and chlorophyll C observed in xanthophyce, bacillariophyce, phaeophyce, chrysophyce and cryptophyce. Chlorophyll D is observed in rhodophyce. Chlorophyll E is seen in xanthophycean species. Fine, right? So these are the algal classes in which different chlorophyll pigments are observed. Chlorophyll pigment types and their classes, important table again. Important table. Next you see is carotenoids. A group of yellow, orange, red, brown pigments. These are also known as protective pigments because they will prevent the plant from photooxidation. Okay, photooxidation. They help in transferring light energy to chlorophyll and they will prevent the cells from photooxidation. We have two types of carotenoids. One is xanthophyll, second will be carotenes. Xanthophylls, 20 kinds of known algae are present. Okay, fucoxanthin is the major pigment in many algal classes. Okay, in many algal classes, fucoxanthin is the major pigment. Carotenoids, these are fat soluble pigments. We have 5 types of carotenoids or carotenes. Those are alpha, beta, gamma, with lycopene. These are different types of fucoxanthin, of which beta carotene is widely spread in the algae. Which is formed? Which type of pigment is widely or mostly widespread in the algae is beta carotene. Clear? Next, below proteins or phycobilins. These are special pigments which will assist light harvesting during photosynthesis. They will assist light harvesting during photosynthesis. They are found only in Cyanophyceae, Rhodophyceae and Cryptophyceae. Okay, so there are 3 types we have. One is phycocyanin, which is a blue pigment. Phenol, and is phycoerythrin, which is the red pigment. And third will be allophycocyanin. We have phycocyanin, phycoerythrin and allophycocyanin. These are 3 types of bilin pigments or phycobilins. Next comes the reserve food material. Pigmentation is clear to us. We are clear with all the types of chlorophyll pigments and in different classes what type of chlorophyll pigment is present. We are clear with carotenes or carotenoids. We are also done with phycobilins. Now it is the time for what? Reserved food material, because this is also one major criteria for algal classification. Reserved food material. Cell wall composition, reserved food material and pigmentation is the major criteria for algal classification. Fine. Now we will move on to the reserved substances in the algae. Final product of photosynthesis is what? Starch. Of course, glucose is stored in the form of starch. The store different forms of food storage is present here. 3 major types of food storage are present. One is polysaccharides, second is fats and third is sterols. So here you see first chlorophyceae and charophyceae. Chlorophyceae and charophyceae. For reserve food is starch, which is similar to that of angiosperms. Both in chlorophyceae and charophyceae, reserve food material will be starch, which is similar to that of angiosperms. Second is rhodophyceae. In rhodophyceae, the stored food is what? Floridian starch. This is widely known to us. Next is cyanophyceae, which is primitive algal species, cyanobacteria. In them, mixophycean starch is present. Mixophycean starch like glycogen is present. Next is phaeophyceae. Phaeophyceae, laminarin and mannitol. This forms the reserve food material. Laminarin and mannitol. Next is chrysophyceae, bacillariophyceae and xanthophyceae. This forms the reserve food material in glucosin. Fine, right? So these are different classes and their reserve food material. Most important table. Just take a screenshot of this one. And then fats. Here we go with this. Fats, they act as reserve food material in many algae. The major use is they aid in the process of buoyancy, floating in water. Okay, those algae which has reserve food material as fats, that will help the algal species to float on the water. They will give the effect of buoyancy, which is the major point to be addressed. In resting cells and reproductive cells, fats are stored as reserve food. Fats are stored as reserve food in both resting cells and reproductive cells. Next is sterols. Sterols are absent in cyanophyceae and they are present in all the other algal cells. Okay, they are present in all the other algal cells. Okay, sterols are unsaturated polycyclic alcohols. Okay, they are unsaturated polycyclic alcohols. Clear with? Most important is polysaccharides, different types of polysaccharides stored in different algal classes. Next is fats and sterols, what are present. And important point to be addressed here, sterols are absent in cyanophycean species. Next about flagellation in algae. Flagellation in algae. Presence or absence, number, type, orientation of flagella are the most important in algal classification. It is most important in algal classification. Here, Cyanophyceae and Rhodophyceae, there is no flagella here. No flagella, Cyanophyceae and Rhodophyceae. Flagella are classified which is based on morphology. We have two types of flagella. One is acronematic flagella, second is pantonymatic flagella. Acronematic, we have smooth surface, non-flimmered type of flagella where the surface of flagella is smooth. Second is pantonymatic, which is tinsel flagella with fine lateral hairs. These are known as flimmers on the surface. The surface will have hairy appendages. The surfaces will have hairy appendages which are known as mastigonemes. These are known as mastigonemes. Here you can see both the types, tinsel flagella and whiplash flagella. Whiplash flagella whose surface is smooth, whereas tinsel flagella will have hair-like structures. These hair-like structures are known as mastigonemes. Clear ma? Fine, right? Now, here we go with different types of flagella. Isocon flagella. These are found in Chlorophyceae where 1, 2 or 4 acronematic flagella are present. 2 to 4 acronematic flagella are present. 1, 2 or 4 acronematic flagella are present. All flagella are of similar type. They are same type. Next, heterocon type of flagella. We have two different types of flagella. Okay, we have two different types of flagella here. You can see flagellar arrangement will be 9+2 microtubular arrangement only because these are of eukaryotic cell. Now we will dwell into the cell structure once. What is 9+2 arrangement of microtubules? What is 9+2 microtubular arrangement in flagella? In flagella, if you see, at the center you have two central microtubules. These central microtubules are encovered by a central sheath like this. Okay, two central microtubules are covered by a central sheath. To this central sheath we have 9 radial spokes. 9 radial spokes. Each radial spoke has 2 peripheral microtubules. Each radial spoke has 2 peripheral microtubules, which is a doublet peripheral microtubules. Okay, so we have 9 peripheral fibrils, 2 central fibrils which have common in motile cells. In Bacillariophyceae, like diatoms, you see in diatoms, pantonymatic flagella is present. This is a major point to be addressed. Mostly you have what? 9+2 arrangement of flagella in all the type of algal species. But pantonymatic flagellum of male gametes, only male gametes will have what? 9+0 arrangement of flagella. Now if you see plastids in algae, okay, we have three types of plastids of course, chloroplast, chromoplast and leucoplast. Chloroplast will contain both chlorophyll A and chlorophyll B with the accessory pigments. They are found in Chlorophyceae, Charophyceae and other eukaryotic species. They are bounded by double membrane. Chloroplast is bounded by double membrane. Next, chromatophores. They contain chlorophyll A. Chlorophyll A is present and accessory pigments will lack the chlorophyll. Accessory pigments will lack the chlorophyll B. Clear till here, ma? Next. Chloroplast position and shape. Here we go with chloroplast position and shape. Chloroplasts, they can be peripheral or central within the cell. Okay. Number of chloroplasts also varies. Like one per cell. One per cell you can see mostly in Cryptophyceae, Parasynophyceae and Chlorophyceae. In these species you can see one chloroplast per cell. These are motile species. And shape of the chloroplast, very important. Shapes of the chloroplast is very important. Here you can see cup shaped Chlamydomonas, griddle shaped Eulothrix, discoid shaped Chara and Vocaria, reticulate shaped Edogonium, ribbon shaped Spirogyra and stellate shaped Zygnema. These are different shapes which are observed in different species of algae. Fine, right? With this we have finished out what? Chloroplast shapes and chloroplast number as well. About thylakoid arrangement and just a second. Thylakoid arrangement and few more components, few more cellular characteristics followed by Fritsch classification and Chlorophyceae. We will deal it in the next class. Till here though it is fine, right ma? Fritsch classification we will do it in the next class, and that's all for today's session. Thank you.